Thank you everyone for allowing me to come here speak today. So title is The Impact of Genetics in Medicine. And this is going to be sort of an outline what I'd like to talk about today. I'm going to give you a brief introduction about basic genetics and two classical approaches in studying genetics. That includes forward genetics and reverse genetics. And I'll get into a little bit more detail on what that means. I'll then talk about the personalized medicine research project, how we're using Marshfield Clinic patient population, that's all linked to electronic medical records, to study human genetics. And I'll give you some examples, some of the research that I'm doing using what's called Phenome-wide association studies, as well as Phenome-wide scan of heritability studies. And I'll give you, again, more detail on what this all means. Then I'll end with sort of the challenges of incorporating this information into genetic medicine. So we have a volunteer here, anonymized, so no one knows who he is, of course. And we're all living organisms, and we consist of cells. And just about every part of our body is made up of cells, including in the bone. And in those cells, we have a compartment that make up the nucleus. And inside that nucleus, we have what are called chromosomes. I'm giving you here an example down here below, hopefully I'm not in anyone's way, of sort of the classical appearance of what a chromosome looks like. Actually, this doesn't, actually this is not how chromosomes normally look like, but except when the cell divides. But nonetheless, it's something that most people can familiarize themselves with. And these chromosomes are made up of long polymers called deoxyribonucleic acids, DNA. And these are the building blocks of what we are, A's, C's, G's, and T's. Now, if you look to your left and you look to your right, you'll notice that, assuming you don't have a twin sitting next to you, that you are all different. And there's a variety of reasons why you're different, and partly that's because of your genetic makeup. So in these long strands of DNA, there may be differences between one spot in the genome compared to another person. And these are called genetic variants. Some of these genetic variants can be extremely rare. Some of them can be extremely common. So maybe in a large population, half of you might have one genetic variant, and the other half might have the other variation. And in these long strands of DNA, we have coding sequence that is what defines protein. It's the functional component of the human genome. And I'm giving you an example here of a gene that perhaps codes for hair color. So you might have a genetic variant that is in this gene that might predispose you to have red hair. Another person might have a genetic variant within this gene that might predispose you to have blonde hair. You could also have genetic variants even outside of a gene that control the gene, regulate it in one way or the other, that might control the intensity of the color, whether if you're light or dark. But more often than not, there are many, many, many genetic variants across the human genome that have no function whatsoever. And my job as a scientist is try to identify where are the functional variants that drive a certain condition or disease in a sea of many non-functional variants, genetic variants. And there are millions of genetic variants within the human genome that makes everyone a little bit different. Now, there are two classical approaches in studying genetics. There's forward genetics and reverse genetics. Reverse genetics is a process. You start with a gene or a genetic variant, and you try to understand what that gene or that genetic variant is doing. So in this case, again, you start with the gene. This thing doesn't work. I'll use a wedge. Should be amazed, because my dad really never took me golfing much, and I still know what this is. So we start with a gene of interest. And the goal is trying to understand what does that gene do, and that's at the phenotypic level, or a condition or a disease. That's what phenotype means. So red hair is a phenotype. Blue eyes is a phenotype. These are all conditions. And this reverse genetic strategy is commonly applied in animal model systems. So you have your favorite gene, and let's give an example of a mouse. So you can use laboratory techniques to literally knock out that gene, so knock the function out. And that's what I'm sort of describing here with the X. And in these animal model systems, you're asking the question, what does this do to the animal? To try to understand what that gene does. And here are some examples of laboratory animals that are used to study genetics in this reverse genetic approach. And here's an example of a mouse that these are its siblings. And you can see this mouse is completely different coat color, even the size is different. So you can knock out one gene and see what happens to that organism, that animal. You can also do this within fish. You can do this within flies. There's a variety of animal model systems to study these genetic conditions. Now, in comparison to reverse genetics, which again, you start with a gene or a genetic variant and try to understand its function by looking at the phenotype, forward genetics is the process of starting with the condition or the phenotype and trying to characterize that condition at the genetic level. So in this case, it's called phenotype to genotype. And you can apply these also in animal model systems, but it's more commonly applied in human genetics. So some of the first human genetic studies that utilize this forward-based approach would include family studies. So many of us are probably well aware that there can be genetic disorders that run in families. So you can take these families and the individuals, look at their genome, and try to identify what are the genetic variants that are causing that condition. So in these family studies, it's typically reserved for rare disease and rare variants. So these are they're called Mendelian diseases, such as cystic fibrosis or a variety of other conditions. And they're usually caused by very rare mutations in the human genome. The alternative to studying rare diseases would be to do association-based testing. So these are reserved for common disease and common variants. And as I had mentioned before, you can have genetic variants that are very rare, and you can have variants that are extremely common. And when you do these association-based testing, you're not necessarily focusing on families, you're focusing on populations of individuals that are presumably all unrelated. And one of the more recent strategies for doing these forward-based approaches in studying common diseases is called the Genome-wide association study. Now, I'll give you a little bit more detail in a second. So this is really supposed to summarize all the genome-wide association studies that have been published thus far. So there have been over 1,000 GWAS, as they're called, that have identified thousands of genetic markers across the human genome. So each one of these lines represents one of the chromosomes in the human genome. Each one of these dots are a genetic marker that's being mapped to the human genome. And each color represents an individual disease or condition. So there's thousands of genetic markers across the human genome that are linked with hundreds of human diseases and conditions. Challenge, though, of these genome-wide association studies is frequently these linkages that have been identified it's very difficult to understand the biological relevance because most of these genetic variants are actually not even in a gene, so trying to understand how that fits into the disease process is a challenge. But also a challenge, which I'll describe at the end of the talk, is that often these don't have any clinical predictive value. They may incrementally increase your risk for a given condition, but if you have a 5% increased risk for a given disease, even though it's statistically significant, it doesn't provide a lot of information when you're trying to apply that to genomic medicine. So I'm going to give you, and again, this is how a GWAS is done. So anyone have blue eyes, raise your hand. Okay. So you are now a case group for blue eyes. Everyone else are controls. And if you have green eyes, I don't know where you fit. So what you do is you take populations, such as this room, and you look at the genomes, everyone that has blue eyes, look at the genomes of everyone that has, let's say, brown eyes, and you scan across the genome every genetic marker that you measure and try to see, is this genetic variant in higher frequency or more prevalent in one group versus the other? And this is called the Manhattan plot. This represents the genome. Each one of these different colors represents a chromosome, and each one of these data points represents an individual genetic marker that's being measured in this genome-wide association study. And then on the vertical axis, we have significance. So again, we measure the first genetic marker and see if the frequencies are different between cases and controls, so we get a significance level, and then we move on to the next one. And we do this 1 million times across the human genome. And the hope is You identify a region on the human genome that has increased significance that might direct you and try to explain why some people have blue eyes and why some people have brown eyes. So that's the whole basis of the genome-wide association study. Now in my work, I'm trying to reverse this paradigm by doing what's called a phenome-wide association study. Okay, so we already asked the question, who has blue eyes, who's got or has had at one point blonde hair? Okay, now I'm gonna ask you everyone else, you can put your hands down. Who's nearsighted? Okay, you become cases for nearsighted. Who's farsighted? You become cases for farsighted. Everyone else is controls. Now we can start getting to more medical relevant conditions. You don't have to raise your hand if you don't feel comfortable, but who has high blood pressure, who's had a history of cancer, who's had a stroke, we can go on and on and on. Many conditions that can be defined in a population is what defines now the phenome. And what I do is I pick a specific genetic marker that I find biologically interesting, and then I ask, is this genetic marker associated with blue eyes? Is it associated with hair color? Is it associated with rheumatoid arthritis, multiple sclerosis, prostate cancer, breast cancer, you name it? And the goal here is to identify no longer the genome, but now I'm trying to identify what phenotypes or what conditions are now linked to my given genetic marker. And that's the whole basis of what I'm doing. So to put these two together, so we have what's the genome-wide association, and now we have the phenome-wide association. Again, in genome-wide association studies, we start with a disease of interest, and we scan the large genome and ask the question what genetic variants are associated with that disease that you're interested in. And a phenome-wide association study, which is, again, a reverse genetic approach, we start with a genetic variant that we're interested in, analogous to that example I gave with the mouse, where you're interested in one gene, you knock it out. In this case, I'm looking at a genetic variant within the human population, and scan all the diseases and ask the question what diseases are associated with that given genetic variant. And this is a pretty novel approach in studying human genetics. So I'm going to give you opportunities to ask questions. So this would be a good chance if anyone has any questions on what I just discussed.
Okay, you said the blonde hair. When I was little, my hair was just as blonde. Got older, it got darker. Now it's gotten lighter. But how blonde to very dark?
I'm sort of, I didn't go dark brown, but when I was born and all through young childhood, I was also very blonde. And then as I got older, it changed color. I don't know the answer to that. There's proteins that define pigmentation, and they might change over time, either based on genetics, but there also could be environmental components to that as well.
Are you looking to see if you knock out a genetic trait? Does it also knock out the diseases or give more opportunity for those diseases to develop, that kind of thing?
Right.
Knocking out that.
So the advantage of doing these phenome-wide association studies is you can link multiple diseases to a common genetic variant. So I'll give an example in a little bit about some of the work that I'm going to show here that describes how a genetic variant that's known to be associated with a given disease is also linked to another disease. So if multiple diseases share a common genetic etiology, then we can learn a lot about both conditions. So one more question, then I'll be able to move on. Please.
Which is easier to do? Here's a disease, now let's study and see what's in common amongst the people who have this disease or condition, or what you're doing?
Well, they both have their advantages and disadvantages. When you go in the forward direction using genome-wide association studies, your diseases are very well characterized because you spent a lot of time and effort to define your cases and controls. but the challenge is trying to get some biology out of this. So you can identify a region, but often these regions are not within genes, and trying to understand the biology is a big challenge. Whereas the opposite is the case. In this case, this is it's called an RS number. This is sort of a catalog-specific marker in the human genome. They all have their own identifiers. And we know something about the biology of this gene. We know something about the biology of this marker. But the challenge now is, how do we define all these diseases? And I'll describe that in a second. But this is a challenge here, is trying to define these conditions, because the information that I'm pulling from, which is specifically electronic medical records, it's not always very clean. And now that's a good segue. So the patient population that I'm studying comes from the Marshfield Clinic Personalized Medicine Research Project, also called PMRP. Now, PMRP represents 20,000 Marshfield Clinic patients. And when they volunteered to be part of the study, they provided DNA samples, which was great for us because then we could measure their genetic information. So of the 20,000 individuals, I'm going to give some examples of data that we created from about 4,300 individuals where we have genetic data. But equally important with all our patients is that they're all linked to Marshfield Clinic's electronic medical record system. Now, Marshfield Clinic is unique for a variety of reasons besides being a decent-sized medical institution in the middle of nowhere. In the mid-'80s, we were one of the first institutions in the United States to develop and apply an electronic medical record system in their normal care. So obviously, you know this is Wisconsin, and it's not very well shown here. There's a light gray region here, which represents the patient population that Marshfield Clinic sees. And this dark gray here represents the patients that were recruited within PMRP, the Marshfield Clinic Personalized Medicine Project. And within our EMR system, electronic medical record system, there's a whole bunch of data that we can pull from it. So we can pull physician notes. We can pull lab values. There's all the biometric measurements like height and weight, BMI, blood pressure, and everything else. But also within the electronic medical record system, we have what's called the IC9 coding system, the International Classification of Disease. In this case, it's version 9, which was IC9 systems the ICD systems are maintained by the World Health Organization. So this is a standardized coding system. And in 1979, we have version 9, and all that data is within electronic medical record system of Marshfield. And the advantage of these IC9 codes is they define conditions. So you go to the hospital, you complain of rheumatoid arthritis, the doctor will treat you and give you a code for rheumatoid arthritis. Send that code to the payer, whether it's Medicare or an insurance company, and that IC9 code has a specific value to it of which will be recovered by the clinic or the hospital. And the IC9 coding system is good because it offers a very wide spectrum of information, going from codes ranging from 001 to 999. Now these codes, everything in between, defines all these different conditions, and they can be grouped together in various ways. At the highest level of resolution, we have what's called a 5-digit code. So I'm giving you an example, 414.11, which defines aneurysms of the coronary artery or coronary vessels. All like codes, so like 414.11, 0.12, 0.13, et cetera, can be lumped into 414.1. All like codes here can be then lumped into 414. And then we can keep going up and up. And at the highest or lowest level of resolution, we have diseases of a system or an organ system. In this case, it's disease of the circulatory system. And what I described here is reiterated down here below in this chart. And what I'm using for defining phenotypes or conditions are these codes right here. And we're using a collapsing strategy so we can actually capture varying levels of information at the disease level. So I talked about those 4,300 individuals that we have genetic data. This represents all the phenotypic information, unique phenotypic information for any given individual within that population. So this is, on the horizontal, are all 4,300 individuals. by unique number of IC9 codes. So on average, each one of our patients have nearly 300 unique codes. That means that there can be a case for 300 unique diseases. And everyone else can be a control if they're not having that specific code. And we have even an extreme example of one patient that has over 800 unique codes. Now to transfer this into a different type of figure, so no longer am I looking at unique codes, now I'm looking at the prevalence, the frequency of each of these codes across the entire IC9 spectrum. So this is 001 to 999, and then we have some incidental V codes. I won't talk about that, but it's also part of this IC9 coding system. And for some codes, they're extremely common. So you go to the doctor and you get your blood pressure measured, you'll probably get a code for that. Most people will have that. Whereas other codes can be more rare, maybe rheumatoid arthritis or multiple sclerosis. And now I hope you can start to appreciate each one of these data points represents a case group and a control group within the same population. And again, this is what's now defining the phenome. So again, I'll give another opportunity for a few questions. Please.
Is there a way to find out our IC9 code?
I know when I go to the clinic, I have they send the billing information to me as well as to the insurance provider, and it has the IC9 codes on it. I don't know if every provider does that. I assume you could probably ask your healthcare institute that you get treated at, but I don't know how that would work. So that's a good question. So IC9 is moving to ICD-10, and IC9 offers 27,000 unique codes. ICD-10 offers 125,000 unique codes. Now, in the long run, ICD-10 is probably going to be better because it allows us to capture a lot more information. So instead of a broken bone, a broken femur, it'll tell you you have a differentiate between left and right femur. There's actually a code that in the ICD-10 system that says that you got in a car accident not once but twice. So there's a lot of information that can be captured with that. For my research, I'm not going to benefit from that coding system anytime soon because all the longitudinal data that we have is on the IC9 system, not the ICD-10. One more question? Ann?
Mine was similar to that. So, when it does expand to the ICD-10, are you going to have to kind of summarize or bring it back down to the level you're discussing?
Yeah, we've been talking about this for a while. If anyone is aware of the Affordable Care, I'm sure everyone's aware of the Affordable Care Act, but one of the rules within the Affordable Care Act was that we'd be transferring over to ICD-10s as of October 1st, I believe, or October 31st of this year. Now, to my chagrin at least, Congress has delayed that at least 1 year. So I'm not worrying about it too much. I'm debating whether or not we're just going to cap on the available information once that transition's been made, or we can retrospectively assess, okay, if you had this code, what would that be translated into an IC9 code? We can do either way, but being that we're still a year plus out, I don't know where it's going to go. So what I'd like to talk about now is how I'm applying all this phenotypic, all these disease condition data that we have within our electronic medical record system to do what's called the Phenome-wide association study. So again, this is the overall strategy of the GWAS, Phenome-wide association study. I'm going to give you some results from 4 biologically interesting genetic variants that have been associated with nearly 4,800 conditions in the 4,300 individuals. And again, the question I'm asking in this reverse process is what diseases are associated or linked to these genetic variants. So these are the 4 genetic variants I'm going to focus on. Again, it's not important to necessarily know what these genes are, but I wanted to make sure you can differentiate between the two. One of the genetic variants that about is because they've already been shown to be linked to a given disease, primarily in the forward genetic, using GWAS, genome-wide association studies. And what I'm trying to do here is demonstrate that if you go in the reverse direction, you can find similar results, as well as potentially gain more insight into the disease processes. So the first genetic variant is again, here's that RS number. Again, not important what it is, but this genetic variant is in a gene called lipoprotein lipase. And in the literature, it's well known that this genetic variant is associated with lipid levels as well as triglyceride levels. Now, when we measure this genetic variant within the 4,300 individuals within Personalized Medicine Research Project, and then link it, or try to link it to the 43 or 4,800 conditions, the IC9 code that defines pure hyperglyceridemia is one of the most significant, the most statistically significant linkages that we identified. So again, something that's known to be associated with triglyceride levels, that's associated with pure hyperglyceridemia, makes sense. So this one's one of our more striking results that we have. Again, if you kind of look at the scale, they're completely different. This only goes up to 4, whereas this one goes up to 10. So this is much more strong. So this genetic variant is in a gene called complementary factor H. It's been well known to be associated with age-related macular degeneration. And lo and behold, the IC9 codes that define AMD are indeed linked to this given genetic variant. Here's another one. This variant is in a gene called HLA-B27. It's an important gene that's involved with immune systems, and if these HLA variants are changed, it often results in autoinflammatory conditions, autoimmune diseases. So this genetic variant is known to be associated with ankylosing spondylitis. So this is kind of like an arthritis of the spine. And when we measure for associations across the phenome, the top association or top linkage signal that we get is for spondylopathies. So again, we're showing what we expect. Now the last example, another variant within these HLA genes, is known to be associated with multiple sclerosis. So in statistics, this would not necessarily be considered significant if you take in consideration all these other linkages that we've measured. So if you flip a coin 1,000 times, there's probably a chance that you're going to get heads 10 times in a row. The more you do it, the more likely you're going to find something that is abnormal than what you would think if you only did it once. So we have this challenge also in genetics, whether you go in the forward genetics with genome-wide association studies as well as in these phenome-wide association studies. So if you flip the coin enough times, you're going to find signals that creep up by chance. Now, this signal is the IC9 code for multiple sclerosis. It's actually, we had only looked at this given code. But what's powerful about this phenome-wide approach is we can actually link multiple conditions, as I mentioned before, to a common genetic etiology. So one of the first studies that looked at this genetic variant going in this reverse genetics approach came out of Vanderbilt. This was a proof of principle study. And one of the incidental findings that they found is the IC9 code that defines erythematous conditions was linked to this specific genetic variant. And what's important is when I went in this reverse approach, is also looking at this genetic variant, we also picked up a linkage with erythematous conditions. So erythematous conditions is a very broad condition defining redness of the face, and a more specific code within that erythematous IC9 code is rosacea. So what's important is that now we can begin to understand, if we understand a little bit more about rosacea, we can gain a lot more insight into multiple sclerosis and vice versa. But also important, if two diseases share a common genetic etiology, then drugs used to treat one disease might be repurposed to treat another. And actually, I'm doing some studies as we speak to try to determine if we have in our electronic medical record system what MS patients are on drugs that are used to treat rosacea, and I'm asking the question, well, what are those drugs doing? Is it changing their disease process? So I showed this slide before, forward genetics in human populations. And as I mentioned, some of the first forward genetic studies used families. And even before we knew anything about DNA, we knew that diseases ran in families. And those were measured by what's called heritability. So what I'm going to talk about now is how we're applying family data. So before it was all unrelated, or at least we're assuming they're unrelated. So now I'm going to talk about how we're going to scan the phenome within families, measuring heritability. So heritability is, so every disease is a composite of genetic and environmental factors, as I already mentioned before about hair color. And the fraction of a condition's variability is the fraction of the genetic variability that causes a given disease is what's defined as heritability. Now, to measure these heritability measurements, the gold standard is to use twin families, but you can also use extended families. And I'm going to talk about how we're using both to try to measure heritability across the entire phenome. So I showed you this map before. Marshfield Clinic sees this region. We have not a complete monopoly, but a fairly good monopoly, about 50-some clinics throughout this region. In our system, we have 2.6 million patients within our electronic medical record system. Now, I can use this information to identify twins. Now, some of you that don't know me, I'm actually a twin myself. And what characterizes myself and my twin sister is that, and for most twins, is we are born on the same date. We are born with the same name. And then there's other information like billing information that, because we're linked to our parents' billing data, could be the same, as well as we're probably coming from the same home address. As well as we might even have documentation within the electronic medical record system that's defined by physicians as actually us being identified as twins. So I'm taking all this information to identify in 2.6 million patients which of them are presumably twins. And when we do this, I identify nearly 20,000 individuals that are likely to be pairs of twins. In addition to the twins, I'm also trying to identify extended families. So when all our patients within PMRP, Personalized Medicine Research Project, were recruited, they filled out a lengthy questionnaire that included asking questions: who are your parents, your siblings, and your children, including all the respected birth dates. We took that information, cross-referenced it with Marshfield Clinic Electronic Medical Record System, and we're able to link multiple individuals within the patient population to many other individuals across the Marshfield Clinic system. So of the 20,000 PMRP patients, nearly 12,000 of them can be linked to another individual within our electronic medical record system, totaling up to nearly 4,500 individual families. Now we can use these families of twins and extended families to measure heritability. Now, what I wanted to do is show proof of principle that we could actually first identify twins and extended families, and then second, prove that we can measure heritability. So I took a very, not very common, but a relatively common heritable disease. It's been well characterized and also has a very specific code within our electronic medical record system. And I focused here on muscular dystrophy. So this is the very specific code. There's actually multiple types of muscular dystrophy. So this is where my benefit when we have ICD-10 systems, where the 5 or 6 different types of muscular dystrophy will actually be broken down into varying levels of the disease. Now, muscular dystrophy is predominantly caused by genetics, and based on the literature, it's estimated that 66% of the disease can be explained by genetics. So when we looked at our twin population, we identified 10 patients of the 20,000 that were identified or coded as having muscular dystrophy. Now, of those 10, 4 of them, representing 2 pairs of twins, were coded both as having muscular dystrophy. And taking this into consideration, when you consider how rare the condition is, and then the probability that we'd have multiple pairs also having muscular dystrophy, this demonstrates that it's unlikely to be caused by anything else but genetics. Now, when we do it in the extended family cohort, we can actually measure quote-unquote heritability. It's a statistical measurement. Of the 16,000 patients, we identified 18 patients coded for muscular dystrophy in this group of extended families. 6 of them come from 3 families, all of them being mother-son combinations. What's interesting about muscular dystrophy is one of the more common forms is it's an X-linked recessive condition. Not very important, but typically if you inherit your, the disease, you'll inherit from your mother's chromosome. That's why often sons will have muscular dystrophy if their mother has muscular dystrophy. And using these families, we can then estimate the heritability of about 0.63, so 63%, which closely matches the 66% previously published. Now, this is completely fabricated data here, but this is what the next goal will be. So using both the twins as well as the extended family, the goal is to measure the heritability for all these conditions so that there are, as I mentioned, there are thousands of diseases. Many of them remain unstudied, and many of them have unappreciated genetic origins. So the goal here is to identify what are the more likely diseases or conditions that are being driven by genetics so we can follow up with those specific diseases. But as a scientist, and in my research project, my goal is to integrate all these approaches, including phenome-wide scan of heritability, which I just talked about. We had the phenome-wide association studies and the genome-wide association studies using reverse genetics with forward genetics for the goal of advancing genomic medicine. So, entertain a few more questions, please.
You haven't said anything about distinguishing between identical and fraternal twins.
Yes. So one of the challenges, well, we can't do that. We don't have the genetic data on the patients that are likely to be twins. So I've been struggling with this for a while. You could use blood type to maybe enrich for twins that are identical versus fraternal. But with the data that we have, we can't separate out the two. What we can do is we compare opposites, the frequencies of these conditions and how they co-segregate within families between individuals that are same sex, which would be enriched for obviously identical twins, compared to opposite sex. So that's one of the things that we're going to be doing. So unfortunately, we can't quote unquote measure heritability within twins because we don't know the genetic relationships, but there's ways that we're working around that. Please.
Suggest that dystrophy but I'm wondering from the fact that these twins have the same.
Yeah. So in this example, we can't truly distinguish the two of them. So one way, again, would be to potentially compare the frequencies of same sex versus opposite sex. So if you see higher concordance or higher pairing of opposite sex, more likely that's going to be due to genetics. So we can't do it with the example I gave because incidentally, they were all boys that were pairs. But because we have so few individuals, it's to have confidence in the results. But my goal is to combine these analyses both within extended families where we can actually know the genetic relationships compared to the twins where the genetic relationship is not necessarily well defined because we don't know who's identical and who's fraternal.
Is there a standard protocol that all genetic centers use in performing these studies, and can you share a database?
Yes and no. So complicated. It's a loaded question, and it's something that the National Institute of Health as well as a lot of other organizations have been struggling with. So if you get funding from the government, one of the requirements to do these types of studies is you have to make this publicly available. But you're walking a fine line is what can you provide that's publicly available? So as a rule, you can't provide anything that links a given individual to the data that you're presenting. And some of the databases that are publicly available just provide a significance level at the population level, so there's no way of defining who has what. Then there's other databases where you actually have to put in a given individual, not necessarily a name or anything like that, but some coded number, so you don't really know who they are, but you actually have their genetic information and then you have their disease information. One of the challenges for my kind of work is that there are a lot of diseases that are all part of a population that's very centrally located. So it's not like the whole United States, it's very specific. So you could actually identify, so we have a code for who's been in a car accident, they have rheumatoid, they had a history of breast cancer. So you can imagine these conditions would cause sort of a fingerprint of who they are. And for many of these conditions, they're outwardly visible, whereas your genetics isn't as. I can talk a little bit more towards the end. I have some of the rules and regulations as an extra slide if the questioning keeps coming up. But we data, but there are a lot of rules and regulations on what we can share and how we can share it. One more question?
Your study is very Northern European.
Yes.
Are you able to compare your findings to other geographic places similar? And are your results similar?
As a rule, they are usually similar, but if they're different, it can be based on differences in the populations. So these genetic variants may be more prominent, exist in higher frequencies within Northern Europeans, and they could be extremely rare within African American populations. But biologically, they might do the same thing. If they're more rare in one population, that means you have less information to measure your linkages, and as such, it's less powerful of analysis. So the best thing you could ever hope for is a very common disease linked to a very common genetic variant. But unfortunately, it's never that. It's always a spectrum of something in between. So the last part of my talk is trying to bring all this information to advance genetic medicine and genomic medicine. And what I like to just mention is the future is here when it comes to genomics and genetics. So for decades now, we've been applying genetics in the medical practice, whether that's measuring our blood types, we have prenatal screening, all these things are typically linked to genetics. Although they may not be measured by the gene, but they could be, again, most of these are all linked to the genetics. Now in 1991, the US government, National Institute of Health, initiated the Human Genome Project. This was finished in 2001 at a cost of $2.6 billion. So this was sequencing every chromosome, nearly every base pair, or those A, C's, and T's across the human genome. That is a significant cost, and again, it took nearly a decade. But now we've had significant advances in technology that now allows us to sequence the entire genome of one individual. So the future is actually to measure the genome. And there are actual clinical institutions, Baylor University, Mayo, a number of Emory, a number of clinics and hospital systems that are applying genomics and actually measuring a patient's entire genome for the sake of standard quality of care. But applying this information has been one of the bigger challenges. And this is for a variety of reasons. One of the challenges is that the biology is not well understood. And as I mentioned previously, there's limited predictive value. I showed you this figure before. There are thousands of genetic variants that are associated with a given hundreds of human conditions. But again, most of these are not in genes, so we don't know what they do. And most of them have very modest effects when it comes to predicting this. So my father allowed me to show his genetic information. And I'm not trying to promote 23andMe, if you know anything about it, but to show how much of a nerd I was or am. 2 Christmases ago, I bought both my mom and my dad a genetic test. Merry Christmas. My kids really wondered why Santa would buy them that. But what they did in this company, so you send in a a swab of your cheek, just a Q-tip. You put it in a little test tube, you send them off, extract the DNA, and then they measure 1 million genetic variants. These are the common variants across the human genome, and then they give you a whole bunch of information about your genome. This can tell you what is your nationality, how much Neanderthal you have in your genome. And incidentally, my dad was at the top percentile. Explains so much. But they also give you some disease information. So my father is at risk for venous thrombolism. He's at increased risk for gout, colorectal cancer, and ulcerative colitis. Now, these are his risks. And this is the average population risk. So he's at a threefold risk of having venous thrombolism. And incidentally, can I share? Okay. He's had this condition. So it might explain. So it might explain, obviously, his genetics. But again, what do you tell a person in the clinic? He said, if you go to your doctor and say, I'm at 35% risk of having a clot, what do I do? And that means you also have about a 65% chance of not having it. What about ulcerative colitis? So he's got a twofold risk of having this condition, but he's only 2.1% chance of having this condition. This is some of the challenges that we commonly run into in trying to interpret all this information. So we have a lot of information, but we don't necessarily know what to do with it.
What did the ancestry show? Where is that?
I'm not providing that, but German or Dutch, I forget. You'd have to ask my dad. But it's actually kind of neat. It's fun. Incidentally, the FDA has stopped them from showing this kind of information because they're worried that they're providing medical advice and they're not licensed to do so. And according to the FDA, this data becomes a device. So they believe they need to regulate it. I have a different opinion on that, but we don't need to get into those politics yet. Now, one of the challenges, again, in addition to trying to understand the biology and trying to understand the predictive value of some of this information, there's also a lot of ethical issues that make to translating this information to make it, to try to understand how to use it. So here's our volunteer again. And not all of this is going to be true, but I hope that it's going to show a point. So this is Mr. H. He's 36 years old. He has an identical twin brother and is married with 2 children. In addition to being a scientist, he works part-time as a school bus driver and Navy combat pilot. Mr. H was recently diagnosed with malignant hyperthermia. And incidentally, that's actually quite common around here. Malignant hyperthermia can be a fatal, it's also preventable disease caused by anesthesia-induced fever. So you have surgery and you're given general anesthesia, your body will react to that and it can be fatal. So there's ways of preventing that, giving specific types of drugs. Mr. H's genome was sequenced. Because it's so cost-effective now to analyze the entire genome, it becomes more practical to do so, but it opens up this can of worms. Typically, you would only look at one gene, but again, the technology is allowing us to do it at the entire genome level. So he had his whole genome sequenced, and they were specifically were looking for mutations in the gene called RYR1. This is the gene known to cause mutations in this gene are known to cause malignant hyperthermia. And incidentally, they identified a mutation within RYR1 that are likely defining his given disease. So one of the challenges is how do we report these findings? Who should have access to Mr. H's complete genomic data as well? Should Mr. H have this data? In this case, it probably would be useful. His doctor, probably useful. What about his family? So he has an identical twin brother. Should he know that? Incidentally, this mutation is what's called dominant, so you only need to inherit one. So that means that one of his parents probably has is susceptible to the condition. That means his children are at a 50% chance of having this condition. Who else do we tell? What about his employer? His insurance provider? And what about the government? So these are some, well, I'm going to give you still unrealistic. This is actually somewhat actually is somewhat realistic, and this is where the conundrum comes in genomic medicine. So in addition, Mr. H is a carrier of 15 known disease-causing mutations. 2 of these mutations significantly increase his risk for Alzheimer's disease and sudden death syndrome. In addition to the 15 known mutations, Mr. H has nearly 50 additional mutations that have no known function, but they're in known disease-causing genes. Now what do we do with this information? So as I mentioned, we can now, instead of looking at one gene, RYR1, because it's so cost-effective, we can actually look at their entire genome. But when we look at their genome, we're opening up a box of who knows what. And actually, most of you are carrying tens of mutations in your human genome that are known to cause disease. But because you don't necessarily inherit both mutations from your mother and father, you don't have the disease. But you have a chance of passing it down to your family. So in this case, who do we tell? Does Mr. H want to know that he's at risk for sudden death syndrome or Alzheimer's disease, 2 conditions you may not be able to prevent? What about Mr. H's doctor? How is he going to interpret all this information, or she? Again, the family. There's a lot more information here to know what to do with. Now the employer. So he could die, Mr. H could die at any minute from sudden death syndrome, and he's responsible for driving 7-year-old children to elementary school every day. Does the employer have a right to know? The insurance provider, again, it's much more complicated. And he's also, as I mentioned, he's a Navy fighter pilot. So he's responsible for a $30 million aircraft that might have nuclear weapons on it. Who knows? Should the government know what his genetic information is? So these are the challenges of the ethical challenges, not only at the biological level, but how we can, the things that I struggle with on a daily basis. So I'll just end with acknowledgments. There have been a lot of people that have been involved in my studies, and I'd like to acknowledge them. I won't list them off by name, but I'd also like to thank the funding organizations that have supported this research. With that, I thank you for your time, and I look forward to any questions you have. Please.
Your research is really broad and sort of encompassing. You don't really design it to target a specific outcome, but when something emerges as interest, are there people that are contacting you or that you're feeding to do further research that's beyond your scope?
Right, so my main research focus is developing these phenome-wide studies. So the phenome-wide association studies I described before, there's only been a handful of publications thus far on the topic, and I've been some of the first ones to publish on this, but it's an emerging field, and then I'm trying to further develop these phenome-wide approaches as I described, measuring heritability across the entire phenome. Now, the advantage of these studies is, again, it doesn't pigeonhole me in studying a given disease, which you're always restricted on your patients. I'm still restricted on the patients, but in this case, I'm trying to use these phenome-wide approaches to identify conditions and diseases that deserve additional follow-up. I gave the example, I wish the significance levels were higher. The multiple sclerosis and rosacea story, which I'm confident is real. I'm, as I mentioned, following up on those signals and trying to understand what's being shared and whether or not we can repurpose drugs across the different diseases. And of course, I'm also following up with collaborations because there's a code for many, many different things, from ingrown hairs to club feet to whatever, and I'm not an expert on any one disease. In the back.
Someone is very careless in identifying these different things. Will that skew your research?
Yes. So I gave the example of going to the hospital. Maybe I did or didn't. In the beginning of, if you go to the hospital, you complain of chest pain, and you get this workup for determining if you've had a heart attack. If they end up finding out that you have angina or some other bad heartburn or whatever, they'll probably still code you as having had a heart attack because they spent a lot of time and effort to determine whether or not you had a heart attack, and they want to recover those costs. So it's not perfect, and some are really good, and some codes are not very good. So muscular dystrophy, it's a very good code because if you get diagnosed with it, you're pretty confident that you actually have it, especially if they follow up with genetic tests. Codes, it can be a challenge.
On these codes, shouldn't the hospital give the patient what code they put in and tell them what it is so the patient can see, is that really what I was here for? They got the wrong one on me. I had a heck of a time getting it straightened out.
I can appreciate that. My hospital, or Marshfield Clinic at least, they give out the codes. So you know what you're being diagnosed with or asked to be charged for. But I don't know if that's standard for all healthcare institutions.
Here, if I understand you right, you're saying that the same code is used for billing purposes and for diagnostic purposes.
They're primarily used, they're mostly used for billing.
Then that really contaminates your research.
It does, yes. So it is not a clean system, but it's the best system we have to define a phenome. Because the billing system, in Europe it would be different, is my understanding at least, what I've been told, is that these ICD codes are more specific to your condition, not necessarily to what you're being billed for. In the United States, we primarily use it for billing purposes, but it does an okay job in defining also the condition. And as an example, by at least 3 of the 4 conditions that I gave those 3 genetic variants, those examples, we're able to pick up the expected association going in the reverse direction compared to what's previously been published. So I'm confident that they do an okay job, but I realize they're definitely not perfect.
Where's the field in using this for drug treatment, using your research? Is it pharmacogenomics?
Not necessarily pharmacogenomics. So pharmacogenomics is a field of study where, based on your genetic code, will dictate on what drug you can have or can't have or how much you can have. So some genetic variants are in genes that metabolize, for example, caffeine. And based on that genetic variant, you might be more or less susceptible to the effects of caffeine. And those genetic variants also transfer to treating prostate cancer or breast cancer. Warfarin is a good example. So I don't know if most of you might know what warfarin is, but there are multiple genes in the human genome that predict how well you're going to respond to warfarin. So from this perspective, we can't necessarily identify pharmacogenetic targets, but what we can do is still link multiple genes to a common genetic variant, and then multiple diseases to a common genetic variant, and if you do so, then you might be able to hypothesize that drugs can be shared between diseases, and we're doing those studies as we speak.
Are there any associations being made for the mental disease genome?
Yes, so I am not limiting to any given disease. So within, I think, right around here are a lot of the mental disorders. Bipolar disease, schizophrenia, and a lot of other conditions. One of the challenges with our patient population, though, is within PMRP is they all had to be able to consent. So if they have any sort of severe conditions, they probably were not in a position to consent. So that's one of the challenges with this patient population. In addition, there's a lot more security when it comes to mental health conditions. So I'm not actually privy to all of the clinical data when it comes to these codes, depending on what department gave them. So if it was in general medical or just like family practice, if that code was somehow made within that practice, they'd probably get the code. But if it was actually in the psych ward or whatever, we probably wouldn't be able to capture that for a variety of regulatory reasons. I don't think so. As my understanding with raloxifene, it's just a different mechanism, and of how it treats the breast cancer. And I should know a lot more about this because the STAR study I think was done at Mayo, if I believe correctly. But I don't believe they've used phenome-wide approaches to do so because right. And with drug discovery, they try to do it intelligently, but often it's just brute force trial and error. So you have these model systems, well, whether it's a petri dish full of cells or it's an animal system, and you give these drugs to these systems and you try to see what it does to a disease that you're trying to study, and you might go through 1,000 drugs to try to find what are the likely drugs to treat this given disease, and then they move on to different stages of trying to develop that drug further. Using this approach, you might be able to streamline that process more when you can link multiple diseases to a disease. So something important that I'm focusing on, oops, this right here. I gave you those two examples of HLA genetic variants that are known to be associated with these autoimmune conditions. If you look at this whole genome, if you try to look carefully, you might find a region in the human genome that's associated with a lot of conditions. And that's right here. You can see all these dots. They're all linked to this one small area, and this is the HLA region. So there are a lot of diseases that have immunological components. And if you start to understand how those diseases are being regulated, then we can begin to treat them in a more intelligent way. Yes?
Is most of what you do on a day-to-day basis crunching numbers from all kinds of studies that have already been done, or do you experiment with the mice or whatever yourself?
Marshfield Clinic doesn't have any animal models facilities, so we can't do any of animal work. But I do a combination of bench work, and I do primarily, so bench work, it might be 10% of my time, and then 90, or I mean, maybe I have 20% time of meetings and everything else, administrative responsibilities, but then the rest of the time is usually writing. So my job is primarily to get grants and to write publications. So we do collaborative research. So Marshfield Clinic has another department that's filled with biostatisticians and bioinformaticians. So these are the math geeks and the computer geeks. And so I generate all the data, the raw data, and then they'll interpret the data and then provide these significant levels where then I try to understand what they mean and publish those results and use that data to get more grants. That's my job.
Grants from where? Pharmaceutical companies or
So
government?
government is my career goal because as a scientist, you want to be independently funded, and you also want to be independent from private interests. So pharmaceutical companies are willing to hand out money, but there's also often strings attached. So as a young scientist, where I'm trying to blaze my own path, I don't want to necessarily be tied to a given company. But there are opportunities through companies. There's also opportunities through foundations, which have less strings attached, but they still, often when you apply to a foundation, like as the Gates Foundation or whatever, Melinda Gates, they often have intellectual property rules. So if you identify something and you can market that something, that foundation might take a certain percentage off the top. Where for me, I focus primarily on National Institutes of Health. This is the primary government agency that provides money for this type of research. But there are definitely a lot of different avenues to pursue. And unfortunately, in this current economic environment, it's not easy either way. Please.
5 or 10 years from now, where do you see us being based, vis-a-vis what you're learning or what you hope to learn? What goes on in medicine?
I believe that the tech so much so that we're not going to care about the biology. We're going to care about it, but the technology is going to go faster than our understanding. And because often our society is driven by economics, we're going to end up probably having everyone's genome sequenced in a decade or so. And the more people that we have genetic information on, larger populations, the better we can understand the biology and better we can understand the etiologies that are going on with any given disease. But as such, my guess is we're going to be moving quicker than our understanding of what's actually going on at the genetic level. One more thing. Has anyone heard of the movie Gattaca? Okay, so it's kind of a cult classic for geneticists. It's got Ethan Hawke in it. It's 15 years old. It's a dystopian movie, so if you ever want to see how bad it can be when you understand genetics, you can watch that movie. And thank you for your time.